Good evening, everyone. So in continuation to the topics that we were discussing yesterday, the aspects that we will first list out the aspects that we'll discuss today, and then proceed from there. So if you remember, we started our discussion about the plant growth, and in that we've already discussed the growth rate, right? And we were actually discussing the arithmetic and geometric growth, right? So we'll start with how the cells actually help in the growth of the plant, that is apical, basal expansion, and this is the first thing that we'll discuss today, and radial expansion as a continuation to the topic that we discussed previously, that's the arithmetic and geometric growth, radial expansion, right? And once we are done, this is going to be a small topic which is left from yesterday, and once we are done with this, we'll move on to the next aspect, which is the conditions for growth. Okay, the various conditions required for the growth of the plants. Okay, then we'll talk about differentiation. What is differentiation? What is de-differentiation? And re-differentiation. Okay, once we cover these three aspects, we'll come to what exactly development is. Okay, in terms of plants, what is development? Okay, and then we'll talk about plasticity and heterophyly. Okay, okay, so these are the various aspects that we'll cover today. Okay, so as you can see, this is going to be a short session when compared to the session that we had yesterday. Okay, so we'll start with the first aspect, which is apical and basal expansion. What is apical and what is basal expansion, and what is radial expansion? Okay, now if you take the plant cells can actually divide in three different planes. Okay, they can divide in three different planes. Now, depending on how they divide, okay, either the length of the plant will increase or the diameter of the plant will increase. Okay, so we'll look at the three planes that are possible. So if you consider each of these cubes as a single cell, okay, each of these cubes as a single cell, the cube can be divided like this in the middle, or like this, or in this plane. So three different planes. This is one plane, this is another plane, and this is the third plane. So you can divide it in three different planes. Okay, now if you divide it in this plane, okay, if you divide it in this plane, you're getting a cell at the bottom and a cell at the top. Okay, when these cells start to elongate and they become bigger, you are actually increasing the length of the plant. Okay, and this, okay, is called apical basal expansion, because you are increasing the apex, that is the towards the top you are increasing, and towards the bottom also you are increasing the length. So this kind of an expansion is called apical basal expansion, where in this plane, when the cell divides, it is actually contributing to the increase in the length of the plant. The other two planes, when the plant cell divides by other two planes, okay, it will basically increase the diameter of the plant. Okay, it will increase the diameter of the plant, not the length of the plant. It will simply increase the diameter of the plant. So the plant will become, if this is the stem, the plant stem will become thicker and thicker. Okay, now this division is called the radial expansion. Okay, so basically what we are trying to say here is, apical basal expansion will lead to an increase in the length of the plant, whereas the radial expansion, okay, radial expansion will lead to an increase in the thickness of the plant. The thickness of the plant will increase when it is radial expansion. Okay, so that's one aspect that you will have to know. Okay, now if we move to the next aspect, which is the conditions for plant growth. Now, if we have to list out all the various factors that are essential for plants, it will be a long list, but we can group them together, and we have a few things which are absolutely essential. We have water, and all the plants require carbon dioxide. They all need oxygen. Then they also need an optimum temperature, right? Optimum temperature for growth. Then of course, in addition to these, they need light and nutrients. Now these nutrients, when I'm talking about, what I'm talking about here is micro and macro elements. Okay, because plants are autotrophs. Plants are autotrophs, so we don't need to give them carbohydrates like human beings or animals, okay, because they can actually make their own carbohydrate, they can make their own food. However, they need certain components like your water, carbon dioxide, light, etc. for making their own food. We'll take water for example. What is the necessity of water in the plants? Okay, I'll tell you how important water is. So if we take water, now water is mainly responsible for the transport. It's like the blood in human beings, transport of molecules. So the various molecules that are transported to the various parts of the plant are being carried through water. Okay, then the next thing is, in addition to the fact that almost 70 to 80% of the cell is actually made up of water. Okay, so water is critical. Now, where else do you see the importance of water? In the various biochemical reactions that occur in the cell, okay, the various biochemical reactions that occur in the cell, you will actually see the requirement of a medium. Okay, you need medium. You can't expect to, let's say, for instance, I'll give you an example. Let's say you want to make a reaction between sodium chloride and let's say calcium oxide, for instance. Okay, you want to create a reaction between these two. You can't expect to put these powders and then expect them to react. They can only react when you actually, if at all they react. Okay, they can only react when you actually dissolve them and bring them into the same medium. So in order to ensure that the biochemical reactions actually occur, you need a medium, and water serves as the medium for the various biochemical reactions. Okay, water serves as the medium for various biochemical. In some reactions, water is critical as well. Okay, in some reactions, water itself will take part, but in most cases, water will actually act as the medium for the various biochemical reactions. Then the other important aspect where you see the water playing a big role is it provides Turgor pressure. What is Turgor pressure? So we know that the plants, if you take some of the plants like your sequoia, right, this grows to almost 106 meters. Okay, so if a plant is growing to such heights, then obviously the cells which are actually forming the base, okay, the base are under tremendous pressure. Okay, they have to, so let's say these are roots. So these cells here, they have to bear a lot of pressure, right? So in order to bear that tremendous pressure and remain, okay, and make sure they are not crushed, okay, because of that pressure, you need an equivalent pressure being generated from inside the cell. So inside the cells, you need an opposite pressure acting on the walls to make sure the cell is not getting collapsed. Okay, the cell should not collapse. If the cell collapses, if all the cells here collapse, the entire plant will collapse. Okay, so the cells here will make sure that they are not collapsing. So in order to do that, an internal pressure has to be created on the walls of the cells here. Okay, and this internal pressure is called the Turgor pressure. And this Turgor pressure is usually provided by the vacuoles. And vacuoles are filled with water. Okay, so water is indirectly responsible for the Turgor pressure because water is actually present in the vacuoles, and vacuoles are the reason why you have Turgor pressure. And without the Turgor pressure, the entire cell will collapse. Since plant is composition of cells, okay, the entire plant will collapse. So Turgor pressure is very critical, and water plays the most important role in maintaining the Turgor pressure. Okay, so what else? And water is also essential for temperature stabilization, right? Why do you think the plants actually release water by transpiration? They release the water by transpiration because they're trying to cool off. Okay, so water plays a critical role in temperature stabilization. Then water is also important for cell elongation and growth. Whenever a cell has to expand, for instance, let's say the cell is like this. Now this cell, before it can actually divide into two, okay, it will first expand first. It will elongate and then divide into two cells of the same size. So this elongation can occur only when there is enough water. Okay, only when there is enough water, this elongation will occur. So the entire space will be filled with water, and that actually helps the cells to elongate. Okay, so water actually helps the cells to elongate, and this is also responsible for the elongation of the cells. Is actually responsible for increase in the height of the plants. Okay, so water is actually contributing to cell elongation as well as the growth of the plants by allowing the cells to elongate. Okay, so that is how critical water is for the plants. Okay, now the various other things that we just listed out, you have carbon dioxide. Of course, you need carbon dioxide for the important process of photosynthesis. Even though I haven't mentioned photosynthesis under water, that is another critical part where you see the requirement of water. Then you see oxygen. Whatever through photosynthesis you are getting your glucose, and in order to burn the glucose and provide and get energy in the form of ATP, okay, in order to burn this glucose that you are getting, you need oxygen. So oxygen is critical. Then what else? Then temperature. Temperature is very critical because most plants die above temperatures of 40 degrees centigrade. Most terrestrial plants will die above temperatures of 40 degrees centigrade. Not for a short period, but for longer periods. If the plants are exposed to temperatures above 40 degrees, there's a good chance that they might die. The optimum temperature for plants for optimal growth is around 20 to 30 degrees centigrade. Okay, and again, this temperature is a factor which is dependent on the region in which the plants are living. These conditions that we are talking about here is for a country like India. Okay, so for most plants in India, it's the optimum temperature is 20 to 30 degrees centigrade. Okay, so it can be different for other regions. Okay, again, the last thing is when we talk about nutrients, the other nutrients which we call the micro and the macro elements, they are all critical because each micro element plays an important role in the overall growth and development of the plant. For instance, if you take magnesium, okay, Mg2 plus, magnesium is involved in many biochemical reactions. In fact, most of the biochemical reactions where you see the energy being consumed, okay, you need magnesium ions. Okay, so we have an elaborate session on the role of various micro and macro elements. Okay, so we'll discuss them in detail in the next few sessions. Okay, so I think it's scheduled in the next couple of weeks. So we'll discuss all these in detail, the various micro and macro elements and what is their importance for a plant. Okay, we'll discuss that in detail. So all these conditions should be met for the plant to grow and develop in the best way possible. Okay, so these are the various conditions that you need for a plant to grow well. Okay, then moving on to the next aspect, which is differentiation, de-differentiation, de-differentiation, and then re-differentiation. These are three different terms. Okay, de-differentiation, re-differentiation, and differentiation, re-differentiation. Okay, so we'll look at each of these aspects. So before actually moving to the, so this is something which I wanted to show you. So this is when the cells actually divide by apical and basal fashion, you see the increase in the length, and the way when they divide in by the radial fashion, that is radial expansion, they increase the thickness of the stem. Okay. Now, before actually discussing these aspects in detail, we'll first see what is the plant actually made up of. Okay, now the entire plant, as you know, is derived from a seedling. You have a seed first, and this seed has an embryo. This seed has an embryo, and we already saw how the embryo will actually grow inside the embryo sac of a seed, and then it finally forms the seedling, right? And from this seedling, you are getting your entire plant. So initially, if you look at this seedling, okay, it is made up of only two kinds of cells: the apical meristems, the shoot apical meristem and the root apical meristem. You can basically say it is made up of meristem cells. Okay, the entire thing is made up of meristem cells. Once the plant grows, whether it is root or stem or leaf, in each of these organs you will see three kinds of tissues. Okay, three kinds of cells can be seen, three kinds of tissues can be seen. The first tissue system is called the vascular tissue system. Okay, the vascular tissue system. The vascular tissue system basically will comprise of xylem and the phloem, and they are involved in the transport of water and food respectively. So the xylem is involved in the transport of water and the phloem is involved in the transport of food. Okay. Then the other tissue system that you see is the dermal tissue system. Dermal tissue system will form the surface of the plant. Okay, whatever you see externally, that is your dermal tissue system. Okay, so all the surfaces are actually covered with, all the surface of the plant is actually covered with the dermal tissue system. Now, between the dermal tissue system and vascular tissue system, whatever is there, that's called the ground tissue system. Okay, so basically you have three tissue systems. Okay, so the plant, I'll get back to this. In order to understand this, you have to first understand the plant composition. So the plant basically have three tissue systems. Any plant, any part of the plant will have three tissue systems. One is your vascular tissue system, right? And the other one is your ground tissue system and the dermal tissue system, right? The vascular tissue system will basically consist of your xylem and phloem, and the dermal tissue system, you can say the epidermis, the outer layers. Okay, the ground tissue system is where you have the parenchyma, mesophyll cells. Okay, this keeps changing. Okay, so in the leaves you have mesophyll cells. Okay, so this is your parenchyma, basically parenchyma, etc., etc. So the ground tissue system will actually fill up the spaces between your dermal and the vascular tissue system. Dermal is the outermost layer, vascular tissue system will be somewhere in the middle, okay, or distributed in the space in between, and then the ground tissue system will basically fill all the other spaces between the vascular tissue system and the dermal tissue system. Okay, so now the ground tissue system will carry out, in the ground tissue system you have cells which can basically carry out photosynthesis, okay, or can store food materials. That's the role of ground tissue system. Okay, and it also supports the plants. It provides the support to the plant. Now what will the vascular tissue system do? The vascular tissue system basically will help in the transport of food materials and water, that we already saw. The dermal tissue system will basically act as a cover and protects the plant from the external environment. Okay, so these three tissue systems are actually coming from a single type of cells. You have to understand that. All the three types of tissue systems that we saw are coming from a single type of cells, which is your meristem cells. So how is that happening? Okay, so here the apical meristem cell, don't worry if you don't, if you can't see the text, don't worry. So this is the meristem cell. So you have your apical meristem cell. This apical meristem cell will give rise to three different kinds of cells. First, it will give rise to three different kinds of cells. Okay, the first kind of cells, so you have your meristem, and this meristem is giving rise to three different kinds of cells. One is protodermal cell, okay, then ground meristem cell, and then you have your procambium. Okay, so from the meristem cells you are getting three cells: the prodermal cell, the ground meristem cell, and the procambium. The prodermal cell is responsible for your dermal tissue system. Okay, prodermal cell is responsible for all the cells that are there in the dermal tissue system are coming from the prodermal cell. Okay, and the ground meristem cell is giving rise to your ground tissue system. Okay, all the cells that are there in the ground tissue system are coming from this cell, ground meristem cell, and all the cells that are there in the vascular bundles, that is your xylem and phloem, okay, are coming from this. So this is vascular tissue system. The procambium is responsible for all the cells in the vascular tissue system. Now, if you take the xylem and phloem, you are very well aware that xylem is made up of different kinds of cells, like your tracheary elements and all, and phloem is made up of different kinds of cells. Okay, by like your sieve tube elements and companion cells and all. Okay, so from a single kind of a cell, you're first getting three different kinds of cells, and these three different kinds of cells are giving rise to many different kinds of cells. So these cells which come from the initial cell, these are called the progenitor cells. Okay, these are called the progenitor or precursor cells. We call them progenitors or the precursor cells. Progenitor or precursor cells. So from the meristem you are getting three progenitor cells, and these three progenitor cells are giving rise to all the different kinds of cells present in the plant. Okay, so here what you have to understand here is the meristem cells and the progenitor cells still have the ability to divide. They can divide. Okay, they can divide. However, the minute they convert into the other kinds of cells, for instance, from the prodermal cells, you are getting your epidermis. Right? From the prodermal cells, you are getting your epidermis. The minute the prodermal cell gets converted into epidermis, the epidermis will no longer have the ability to divide. It loses its ability and it performs just one function, which is to form a covering and protect the plant. So here we are starting with a cell which can actually divide and transform or convert into any kind of cell. Okay, so here let's take this example, the procambium cell. The procambium cell has the ability to convert into xylem tissue or the phloem tissue. Okay, let's just so before actually going into those details, let's see. So from the ground meristem cell, you're getting the parenchyma cell, the collenchyma, right? Collenchyma and the sclerenchyma. sclerenchyma. All the three you're getting from the ground meristem cell. Okay, now what are we getting from the proderm cell? From the proderm cell you're getting the pavement cell, okay, which is also present in the outer layer itself. You're getting your guard cell, that is actually guarding your stomata, and then you're getting your the hairs which are present on the plant, they're called trichomes. You're getting those trichomes as well. All the cells that you actually find on the surface are being derived from, in addition to the epidermis, are being derived from your proderm cell. Okay, then from the procambium, what are you getting? You are getting your xylem and phloem. All the xylem elements and the phloem elements are coming from the procambium. Okay, now what you have to understand here is, from the same cambium cell, from the same procambium cell, you are getting both the xylem as well as the phloem. Okay, the xylem element and the phloem element are being formed from the same cell. Let's see how that happens. Okay, you have your cambium cell, right? The cambium cell. The cambium cell will first divide, right? It is dividing, it's growing in length and it's dividing into two halves. One cell will now become the xylem. Okay, one cell will now become the xylem. Then the next one half will still remain as a cambium cell. Okay, so this is, if you remember yesterday's lecture, this is arithmetic division, where one cell is actually converting into something and the other cell is actually remaining in the original state. Now what will happen here? This cambium cell is again dividing, and now the other half is forming the phloem. Okay, from the same cambium cell you are getting your xylem and you are getting your phloem from the same cambium cell. Okay, this cambium cell is dividing. One section towards this side, it is actually forming xylem elements. All the cells which are actually present on this side are forming the xylem components, and all the cells which are present on the other side are forming the phloem components. So what is important here is, from the same cambium cell you are getting your xylem as well as phloem. Okay, however, once it forms xylem, this cannot go back to form the cambium again. Okay, it cannot go back to form the cambium again. In the same way, the phloem cannot come back and form the cambium cell again. So these cells are committed. Okay, they have a purpose now. The purpose of the xylem is to transfer water. Although both are coming from the same cambium cell, they have different purposes, they have different functions. Xylem function is to transport water, phloem function is to transport food materials. Okay, so if you have to define differentiation, define differentiation, right? So differentiation is a process by which, right, it's a process by which a cell acquires, okay, structural, functional and metabolic properties not or that are distinct or different from its progenitor. Okay, so what do we mean by that? The cell which is actually formed from its progenitor is now different structurally, functionally and metabolically. Okay, so its metabolic reactions are different, its structure is different, its function is also different. The cambium function, okay, was to divide and form new cells. Once the new cells were formed, they got transformed into xylem cells. Now the structure of xylem cell is different from the cambium. The function of the xylem cell is to transport water, so it is different, and the metabolic reactions which occur inside the xylem cells is different. In fact, they don't even have the protoplasm. Some of them don't even have the mature cells don't even have the protoplasm. Okay, so if you have a cell and this cell, okay, is undergoing transformation, so you have cell A, now cell B is formed, okay, and this cell B can no longer divide, this can no longer divide, no longer divide. Okay, and then it has different structure, different function. Then we say that the cell A has differentiated into cell B. Okay, it can no longer divide. It has a different structure, it has a different function. Then we can say that cell A has now differentiated into cell B. Okay, so that is what is happening here. So we have your meristem cell. That meristem cell is giving rise to 3 different cells, and these 3 different cells are giving rise to many different cells. So once from the ground meristem cell, once the parenchyma cell forms, we say that it has differentiated. Okay, in the same way, from the procambium, when the xylem or the phloem forms, okay, when the from the procambium, okay, we saw this, how the cambium cell is actually dividing to form xylem and or phloem here. Okay, we say from the cambium xylem formed, and this xylem is a differentiated cell. This is a differentiated cell because it cannot go back to form the cambium again. Okay, so once it loses its ability to divide, it loses its structural and functional and metabolic properties. That means when I say it loses, we are talking about from the original parent. Okay, if the properties, the structural, metabolic and the functional properties is different from the original parent, we say the cell is differentiated. The cell has changed. Okay, so that is about differentiation. So you can see this differentiation in almost all kinds of organisms. For instance, if you take human beings, even human beings, I told you this example even yesterday. From the stem cell, you're getting your progenitor cells. Basically, you're getting your myeloid, progenitor, and lymphoid progenitor. And these cells are actually responsible for all the blood cells in the body. So from these 2 kinds of cells, you're getting all the different kinds of cells. So basically what is happening, these cells are differentiating to form various kinds of cells. Okay, so that is about differentiation. Okay, now we'll move on to the next aspect, which is de-differentiation. What is de-differentiation? Okay, now de-differentiation is where so you have a cell A. Okay, this cell, right, this cell is getting converted into cell B. Okay, now this cell B can no longer divide, and it has different characteristics. Okay, characteristics from parent cell. Okay, it's supposed to be progenitor cell. For simplicity, I'm writing it as parent cell. Okay, then we say this is a differentiated cell. Okay, this is a differentiated cell. For instance, let's take this example. Okay, so you have your cell. Okay, it is undergoing many changes. You don't have to worry about the text here. It is going many changes. It is losing all its internal components. You see, the central portion is your vacuole. Then you have the cytoplasm surrounding it. The pink portion is your cytoplasm surrounding it. So it is losing all the components inside. Okay, and then finally forming the tracheary elements, which is a component of your xylem. Okay, so this is a differentiated cell. This is the original cell, and this is the differentiated cell. Now once it gets differentiated, there's no coming back because it has lost all its contents. It cannot come back to the original form. It remains in the differentiated form. However, some cells can actually come back to a phase where they can divide. They retain the ability to divide. Okay, they retain the ability to divide. Okay, so the differentiated cells, in the example that we saw where trachyary elements was forming, there's no going back. In such situations where you lose all the internal contents of the cell, the nucleus, the protoplasm, etc., there's no coming back. However, if you take the example of parenchyma, okay, if you take the example of parenchyma cells, okay, parenchyma cells, we've seen here that from the ground meristem cell, you are getting your parenchyma cells. From the ground meristem cells, you are getting your parenchyma cells. These parenchyma cells, under certain conditions, can get converted into a different kind of a cell. That is the xylem or the phloem. Okay, has the ability to get converted into this. And in order to do that, it will first get converted into meristems. It will first get converted into meristem. Okay, and the meristem will then again give rise to your xylem or the phloem. So the meristem here is your cambium. So here, when a cell, okay, reverts back to its original form, we say the cell is de-differentiated. Okay, so the parenchymal cells can actually de-differentiate, okay, to form intervascular cambium or cork cambium. Okay, intervascular cambium or cork cambium. Okay, so here let me just show you a figure. So here you can see this region over here in the middle is called the intervascular cambium. So this is your xylem and phloem. These are your vascular bundles. The pink and the blue ones are your vascular bundles. Okay, between the vascular bundles you have intervascular cambium. Okay, now this intervascular cambium, okay, can come from the cambium tissue, which is actually coming from the meristem. From the meristem you are getting your cambium, right? And from the cambium you can get your intervascular cambium and vascular cambium. Together we call it vascular cambium. Okay, so this is cambium or vascular cambium is the same thing, right? However, in some situations you can actually see the ground tissue, that is your parenchyma. The parenchyma cells have the ability to form the cambium. Okay, they have the ability to form the cambium. Now how do they do it? The parenchyma cells will first convert into the meristems, which are your intervascular and okay. So that is how from here on they can actually form your the xylem and phloem. Okay, they can convert into cambium, and from this cambium again xylem and phloem can be formed. Okay, so this is de-differentiation. So a cell which is actually committed, which is differentiated, going back to its original form, is called de-differentiation. Is going back to original form and is called de-differentiation. Now, so here you can see here. So this is a parenchyma cell, okay, which has lost its ability to divide. Okay, now it's actually going through changes. It's going through some changes, and finally it has obtained the ability to divide in 2 days. Okay, so now this has become the cambium. Okay, so this conversion where a cell which is not able to divide is now converted into a cell which is able to divide is called de-differentiation. It's called de-differentiation. And this de-differentiation is a common aspect you will actually find in tissue culture. Okay, the reason why you pick up any tissue from the plant, okay, except for the tissues which have lost the the nucleus and the protoplasm, like I said at the beginning, that's an exception. If there's no nucleus or the protoplasm, then you can't expect the cell to divide. You can't expect the cell to go back to its original form. Okay, so as long as a cell has a proper nucleus and tissue, if you get any protoplasm, if you get any tissue from the plant, keep it in proper hormones and provided conditions, okay, necessary for the growth, it will start growing and it will grow into a complete plant. The reason why it can do that is plant cells have the ability to de-differentiate. This is a unique phenomenon where a differentiated cell can go back to its original progenitor form and start dividing again. It starts to divide again. So this will start dividing again. Okay. Now, after a few divisions, okay, after a few divisions of de-differentiated cell, okay, after a few divisions of de-differentiated cell, what will happen is the division will stop. Okay, the division will stop, and this division, when it stops, the cells are again getting differentiated. Okay, they're getting differentiated again. So what did we start with? We started with a cell which is meristem, that is a pluripotent cell or totipotent cell. We started with a cell which is totipotent, that is, it has the ability to divide and it has the ability to convert into any form. A cell which has the ability to divide and convert into any form is called a totipotent cell. We started with the meristem which is totipotent. This meristem has converted into a progenitor, and this progenitor gave to a cell which is now differentiated. Now, for some reason or under some conditions, the differentiated cell has undergone changes and it has de-differentiated. Okay. Now, after de-differentiation, it is going through divisions again. Okay, you're getting many cells. And after a few rounds of division, these cells stop division. And when these cells stop division, we call them re-differentiated cells. So they were differentiated before, they got de-differentiated, and these de-differentiated cells again went through division and then again differentiated. So it's not the first differentiation, it's the second differentiation. So we call it re-differentiated cells. Okay, let me just show you the entire thing once. Okay, so this chart will actually make it clear. So you have a cell. Okay, this cell is basically equivalent to your meristem. Okay, this is a totipotent cell which can actually divide, okay, and form new cells, and these cells can be of any kind. Okay, so this cell is actually moving forward, it's getting differentiated, right? You're getting a differentiated cell. Now the differentiated cell, once it gets differentiated, it has two options. One is go back to its original form. If it is going back to its original form, this is called de-differentiation. Or okay, once it gets committed, if it's going back to its original form, it's called de-differentiation. Okay. However, it can, once it de-differentiates, it can start dividing. Okay, it can start dividing to form more cells, which after some time will stop dividing. And when they stop dividing, we call it re-differentiation. So a cell, a totipotent cell will first differentiate. The differentiated cell will again go back to its original form, okay, and then again start to divide. When it again starts to divide and stops dividing, we call it a re-differentiated cell. Okay, now the other fates that a differentiated cell can have, is it can go to death, it can die, it can simply divide, forming more and more cells of the same kind. It can continue to divide and form more and more cells of the same kind. Okay, so this de-differentiated cell have two fates: form more cells, and some of the cells will stop dividing. When they stop dividing, we call them re-differentiated cells. Okay, so that's the difference between your differentiation. So basically I'll explain re-differentiated one last time. So you have a meristem cell. This meristem cell is dividing and forming new cells, and these new cells are getting differentiated, that is committed to one form. Okay, however, some of the cells under some conditions can de-differentiate, okay. After they de-differentiate, they will again continue to divide, okay. After a few divisions, okay, what will happen? Cells stop dividing, and these cells which stop dividing are said to be redifferentiated. Okay, these cells which, so redifferentiation will only occur after de-differentiation. Okay, that's the basic difference. Redifferentiation will occur only after de-differentiation. Okay, only the cells which have de-differentiated will give rise to redifferentiated cells. Okay. Now, so yeah, this thing that we see, the redifferentiation can be seen mainly in your tumor cells. Okay, the cells will, the first you have your cells which are going through de-differentiation. Once they de-differentiate, they continue to divide, and after getting a few cells, okay, some of the cells are actually committed to one form, and we call them redifferentiated cells. You can see them in the tumor cells. Okay, now the other aspect that we have to understand here is, so basically everything is starting from a, in a plant, everything is actually starting from a meristem cells. And this meristem cell can take any of the parts. It can become the dermal tissue, it can become the ground tissue, or it can become the vascular tissue. So the fate of this meristem cell is not fixed. The fate of the meristem cell is not fixed. Okay, so here this is called open differentiation. The term which is used is open differentiation. So this is open for any kind of differentiation. The meristem cell is open for any kind of differentiation. That is, it can go and form dermal tissue. Okay, in the dermal tissue it can form a guard cell, it can form a trichome, anything. In the ground tissue it can form a parenchymal cell, it can form a sclerenchymal cell, it can form the collenchymal cells, and in the vascular tissue it can form the xylem or the phloem. So it is open for every option. So that's the reason why in the plants we see open differentiation. Okay. Now, how do they actually, so if it is open differentiation, how do they actually commit to one kind? So for instance, you have your root apical meristem. I showed you the diagram yesterday. So you have your root apical meristem. So tip of the root have many cells which are actively dividing, which we call the root apical meristem. Right, we have the root apical meristem, and below that, if you observe, below the root apical meristem, you will actually see the root cap. Okay, now the meristem cells which are actually dividing here in this portion are pushed down. Okay, they are pushed down to form the root cap. All the cells are differentiated into cells of the root cap, and the cells which are actually present towards the border, okay, will actually form the epidermis. So what we are trying to say here is differentiation will depend on two aspects. Okay, whether a cell will convert into one form or the other will depend on its structure. Okay, what structure it will get finally, and the other aspect is location. Where exactly is it present? For instance, in the example that I just discussed, if it is present towards the bottom of the root apical meristem, it will simply form the root cap. If it is present towards the wall of the root apical meristem, it will form the epidermal cells. So the location and the structure that it finally takes will decide what will be the nature of the differentiated cell. Okay, so if it is close to the tip, it will be pushed towards the root cap and it will form one of the root cap cells. If it is present towards the wall, it will form the epidermal cell. Okay, so the location is critical. What path it will take will depend on the location of a particular cell. Okay, now, so we'll look at the definition for development. Okay, how do we define development? So development, if you have to define for any organism, it is all the events that occur from basically from birth to death. All the events that occur from birth to death, we can say it is development. Okay, so here if you take the plants, we are talking about germination to senescence. Germination to senescence. Senescence is nothing but aging. So finally the plant will deteriorate towards the end. Okay, so all the events that occur in the gap is your development. Okay, so here basically if you look at the development in development of a plant cell or a plant organ or a plant tissue, okay, it will go through several stages. A plant cell, organ or a tissue will go through several stages. Now, what are the stages? Okay, so everything will start with meristematic cell, like I said in the beginning. Everything will start with a meristematic cell. This meristematic cell will increase in its length, that is plasmatic growth. It will increase in its, the growth of the cell will happen. Then it will divide. One cell will undergo differentiation, which we have seen in the cambium example. One cell is going through the differentiation and one cell is coming back to form the meristematic cells. Okay, now when this is going through the differentiation, you will see the overlapping. So elongation and maturation will overlap with the differentiation process. So the differentiated cell will finally mature. Okay, the differentiated cell will finally mature, and the mature cell, after living for a few days, months or years, depending on what kind of a plant it is, what kind of a cell it is, it will finally reach the death. And this process, okay, is what we call the senescence. Okay, where the death in plants will not occur like in animals or in human beings, where you know you are alive 1 minute and the next minute you are dead. Okay, it's not like that. Death in plants is a very slow process. Gradually, from one part of the plant, everything will be taken away. Okay, for instance, when a leaf has to drop from a plant, or when a plant has to get rid of its leaf, the first thing that it will do is it will start sucking out all the nutrients from the leaf. Okay, so it will start gradually sucking out all the, it will first cut off the nutrients supplied to the plant, and whatever nutrients are there in the leaf already, it will start sucking in gradually. Okay, so the leaf will finally, that process, that's the reason why you see the leaves changing color and gradually falling off the plants, and that entire process is what we call the senescence, whether it is a leaf or whether it is a fruit. Okay, so the entire process is called senescence, and finally it leads to a death of a particular tissue, organ or a cell. So this is the various, these are the various steps through which a cell or a tissue or a plant or an organ goes through in a plant. You start with a cell which is totipotent, basically has the ability to divide and transform into any kind of cell. This divides to form a new cell. That cell differentiates. Now it is committed. It matures finally. The mature cell will undergo senescence process and finally it will die. Okay, so these are the various steps that you will actually see in the development of a particular plant cell, plant tissue or a plant organ. Then I have to explain the last two aspects, which is plasticity and heterophily. Okay, now one important thing that you have to understand here is plants adapt very well to the conditions. Okay, plant adapt very well to the conditions. For instance, so depending on, I'll give you an example. Depending on environmental conditions, they change their characteristics. Depending on environmental conditions, they change their characteristics. So they are not fixed. Okay, they can mold themselves in any way. That's the reason why they are said to be plastic. Okay, plasticity is the ability to, so just like plastic, plastic can be, you know, you can just heat it for a minute and you can mold it in any fashion you want. Okay, basically that's plasticity. You can give it any shape that you want. So plants are also like that. They can, you know, make the changes according to the environmental conditions that we provide them. Okay, so they make different kinds of changes to adjust themselves to the environmental conditions being provided to them. Okay, so what do I mean by that? What do I mean with it? I'll just give you a few examples and we'll close this session once we are done with the examples. So let's take a plant, okay, which is actually growing in high carbon environment, high carbon dioxide environment. So when you have enough carbon dioxide, okay, you don't need more stomata because the carbon dioxide will ultimately enter through the stomata. The carbon dioxide required for the photosynthesis process will enter through the stomata. However, if a plant is actually growing in an environment where there is low carbon dioxide, when the carbon dioxide is low, it needs to, you know, increase the intake of carbon dioxide. More and more carbon dioxide has to be taken because already in the environment very less amount of carbon dioxide is available. So in order to accommodate more carbon dioxide, in order to take in more carbon dioxide, the plant will start making more stomata. You can see here, it's actually the same plant is making more stomata. Okay, so it is basically increasing the stomata density in order to accommodate the environmental conditions. Okay, in order to accommodate the environmental conditions, it's actually increasing the number of stomata. Okay, so this is plasticity. Okay, bend yourself according to the conditions provided to you. Okay, this is plasticity. It can actually change according to the conditions being provided to it. Okay, now what is heterophily? Heterophily is where different plants, the same plant produces different kinds of leaves under different conditions. Okay, I'll give you an example here. Okay, now, so this plant over here, this plant, the name of this plant is Roripa aquatica. Okay, this plant, when you grow this plant at a temperature of 20 degrees centigrade for 1 month, the leaves will be like this. The leaves, you can see the picture here. The leaves will look like this. At 20 degrees centigrade, if you grow this plant for 1 month, the leaves will have this form. However, at 30 degrees centigrade, if you grow them for 1 month, the leaf will be like this. Okay, so this is a part of plasticity. Okay, depending on the conditions, the plant is actually adapting and changing its form, changing its characteristics. Okay, so here the change is actually occurring in the leaves. So this change, or in other words, the plasticity that occurs in the leaves, okay, the change that occurs in the leaves is what we call the heterophily. Okay, heterophily. So you can see another example here. This is Arabidopsis. Okay, Arabidopsis. Here this is under low light. This is how the plant grows, the same plant, and in near light it grows like this, and it is in normal light, sorry, in normal light it grows like this. In high light conditions the growth will be something like this, and in normal field conditions outside, these three are in the lab, and in normal field conditions the growth will be something like this. Okay, so the plant behaves differently to different environmental conditions and changes its characteristics according to the environmental conditions being provided to it. Okay, so another example that you can see here is, yeah, this is another example where the factor here is also light. Okay, so here when you go, this plant is called passiflora. passiflora. Okay, when you look at the leaf of this passiflora plant, this is when you give 50% of sunlight, 50% of incidence sunlight, 50%, so partial light. When you give it 100% sunlight, this is what happens. 100% incidence sunlight. And when you wrap, a porous paper, so the regions where you are not actually seeing, these are holes. So perforated paper, when you take a paper which has holes and wrap it around the leaf, you will see those regions in which you're getting sunlight, direct sunlight, they are turning the purple color or pink color, and the remaining regions where you're getting less sunlight is remaining green in color. So depending on the environmental conditions, the plant can actually change its form. Okay, and this is what we call plasticity. And again, when I say environmental conditions, the soil is also another factor. So you can see these are hydrangeas. Hydrangeas. Okay, so here these are flowers which you will find in Southeast and Southeast Asia mostly. So these, depending on the pH of the soil, you'll see different colors of the the same plant forming different colors. Okay, so this is when you grow the plant at pH 4.5, it will basically give you blue colored flowers. However, when you increase the pH of the soil to 7, it will start making pink colored flowers. Okay, so this ability of the plants to basically change according to the environmental conditions is what we call plasticity. And if the change occurs in the leaves, we call it heterophily. Okay, so that's all for today's session. So with this, we have actually finished the plant growth and development chapter. So next time when we meet, we'll discuss a new chapter altogether. So yeah, the questions which were asked so far are: what type of cells are meristem cells? Meristem cells are, you can call them totipotent cells, totipotent or pluripotent. Okay, the cells which have the ability to divide, okay, and transform into any kind of cell. Divide and transform into any kind of cell. So that is your meristem cells. Okay, so these meristem cells are like the stem cells of your human body, are equivalent to the stem cells of your human body. So all the cells in our body are derived from a single cell which we call the stem cell. Okay, so these meristem cells are equivalent to the stem cells of your human body. Yeah, so the next question is, what is the reaction for oxidation in plants? Basically, oxidation is wherever you see the breakdown of a particular molecule, whether it is glucose or fatty acids, if they are being broken down to carbon dioxide, okay, all the reactions that occur in between are oxidation reactions. Okay, so wherever you see the burning of these fuels to release energy, we say oxidation reaction is happening. Okay, so that's all for today's session. Keep following us on Facebook, Instagram and YouTube channels. Subscribe to our YouTube channel and try our app. Do download it. So until we meet next time, bye for now.